today's lecture we are going to discuss the sexual reproduction in flowering plants. Flowering plants, they produce a special modified structure for sexual reproduction, and that structure is called as flower. Now in the diagrammatic representation, here it is as a flower. The flower is the highly modified and condensed shoot on which the floral parts are arranged in circles, and those rings or those circles are ideally called as whorls. In a typical flower, there are 4 different types of whorls, and those are the outermost and the one which is protective and photosynthetic in function is called as calyx, and calyx is made up of sepals. The second one is beautifully colored, sweet scented and attractive, and that is called as corolla, and corolla is made up of petals. The third whorl which is present inside the corolla, and that is called as androecium. This androecium is the group of stamens, and the last whorl is gynoecium. This gynoecium is made up of single or many carpels. So these are the 4 whorls present in a typical flower. Now let us discuss the role and actual condition of these whorls. The outer whorl, calyx and corolla, these are not directly involved in the process of sexual reproduction, but they are supporting the process of sexual reproduction. Calyx is green in color and protective in function. It is protecting the internal part of the flower in bud condition. Corolla, it is beautifully colored as we have discussed and is responsible for attraction of the pollinators and helping the process of sexual reproduction. And therefore, these 2 whorls are called as accessory whorls. While androecium and gynoecium, these 2 whorls are actually directly involved in the process of sexual reproduction, in which androecium is the male reproductive part and gynoecium is the female reproductive part. And these are the gonads of the flower. They used to produce the male and female gamete respectively, and thus these 2 whorls are directly involved in the process of sexual reproduction. So androecium and gynoecium, they are ideally called as essential whorls. So in a typical flower, 2 whorls are accessory whorls, those are calyx and corolla, and androecium and gynoecium, these are the male and female reproductive structures respectively. They are called as essential whorl. They show direct involvement in the process of sexual reproduction. To study the complete mechanism of sexual reproduction in flowering plant, you must know the life cycle of the flowering plant, what we call it as heteromorphic alternation of generations. This part you have already studied in the topic Kingdom Plantae in your 11th standard syllabus. The heteromorphic alternation of generation in the plants is related with 2 different generations which are coming one after the other in the life cycle of the plant. As we are discussing the sexual reproduction in flowering plants, they show 2 distinct generations in their life cycle. The first one is called as sporophytic or sporophyte generation, which is related with spore formation, and this generation, this stage of life cycle is diploid. The second phase is gametophyte, and how it is developed, let us see in the life cycle. Now sporophyte is used to produce the spores. One is called as microspore, another is called as megaspore. Both are haploid because these are the product of meiosis. So sporophytic plant undergo meiosis to produce the microspores as well as megaspores. Sporophyte is the actual plant of flower or the flowering plant. The plant which you observe by your naked eyes in case of angiosperm and gymnosperm is sporophyte, which is diploid, which will undergo meiosis in their gonads to produce microspore and megaspore. Now microspores and megaspore, they germinate and develop into male gametophyte and female gametophyte. these are the haploid structures because they are produced from microspore and megaspore respectively. Now male gametophyte and female gametophyte, these are the structures which are actually producing the gametes. Male gametophyte will produce the male gamete. Gametes are obviously haploid. Female gametophyte will produce the female gamete, which is also haploid. Now, these gametes will come in contact with each other and they show a process which is called as fertilization. So, after fertilization, male and female gamete fuse together to form a structure which is called as zygote. And zygote is diploid in nature because it is developed by fusion of male and female gametes. This zygote divide and redivide by mitosis to produce an embryo, and this embryo germinates to develop the new sporophyte. In this way, the sporophytic phase and the haploid gametophytic phase, they are coming one after the other in the life cycle of the plant, which is called as heteromorphic alternation of generation. Now let us split this diagram into two, and that is, in one part all the diploid stages are included. Embryo, it is again diploid, zygote is diploid, sporophyte is diploid. Now just draw a line like this. Now this part of the life cycle is diploid and this phase is called as sporophytic phase. Now see, observe the rest of the phases. These are haploid, means spores, gametophytes and gamete. Now this part of the life cycle is called as gametophytic phase and it is haploid in nature. Now this is called as the heteromorphic alternation of generation, or simply you can consider it as life cycle of the plant. On the basis of this diagram, on the basis of life cycle of the plant, we can summarize the steps which are involved in the process of sexual reproduction. Let us discuss one by one. The first step is so while plants are proceeding for sexual reproduction, they must firstly form microspores as well as megaspores, and therefore the first step is called as Microsporogenesis. It means formation of microspore. This microsporogenesis occur in stamens, in androecium as we have discussed, and because it is the male reproductive structure. The second phase is called as Megasporogenesis. In this process, megaspores are formed. Megaspore genesis means formation, so megaspores are formed, and this will occur in the ovule of the gynoecium or of the carpel, and therefore it is called as female reproductive structure of the flower. So these two steps are there, microsporogenesis and megasporogenesis. Now the third one is itself, it is indicated in the diagram, development of male and female gametophyte. We can collectively place in one point, and that is third, development of male and female gametophyte. Now these gametophytes will further produce the gametes. Now these gametes should come in contact with each other, what we call it as fertilization, for development of zygote and later stages of the embryo. So in advanced plants like flowering plants, angiosperm and gymnosperm, female gamete never leaves its place. It will remain as it is in the mother plant, or at its place male gamete has to travel up to the female gamete, and the traveling of the male gamete up to female gamete is also called as pollination, and therefore the fourth step in the sexual reproduction of flowering plant is called as pollination. After pollination, the next step is fertilization, obviously, and therefore the fifth step is called as fertilization. And lastly, after fertilization, the ovary is converted into fruit and the ovule is converted into seed, which is called as post-fertilization changes. That is nothing but development of zygote, embryo, endosperm, etc. We can collectively mention in a single point, and that is the last point, and that is called as formation of fruit and seeds. So in this way, you can summarize the entire process of sexual reproduction in the plant in 6 different steps. First is microsporogenesis, which will occur in the anthers or stamen or androecium, whatever it may be, but it is the male reproductive part, you must remember this. Megasporogenesis occur in the female reproductive part, especially in the ovule. Then the third step is development of male and female gametophyte, then pollination, then fertilization, and lastly formation of fruits and seeds. Let us discuss the first step of sexual reproduction in flowering plant, and that is microsporogenesis. Now, firstly we have to discuss what is the site of microsporogenesis. It will occur in the structure which is called as androecium, or specifically you can call it as in stamens. A stamen is the individual part of androecium, and it is involved in the process of microsporogenesis. Now, the structure of stamen is like this. It consists of 2 parts. One is called as anther, and the second one is called as filament. Out of these 2, the fertile part of the stamen is anther, and which is related with the process what we call it as microsporogenesis. So the site of microsporogenesis is, you can call it as anther. If it is there in option, it is stamen and androecium, both are the correct answer. But when anther is specifically given, then your answer must be anther. Anther is the fertile part where microsporogenesis occurs. So first we have to study the structure of anther before studying what is microsporogenesis. For that purpose, we have to cut the anther in cross section. So we are going to take TS of angiospermic anther. See, when you cut the angiospermic anther, basically anther is a structure which is bilobed or dithecous. It is having 2 lobes, so it is a bilobed structure. When you cut the anther, it will show you 2 lobes like this. This is the first lobe, and this one is the second. These 2 lobes are attached with each other. Now at one point you get the connective attachment, because filament is attached to the anther by a structure which is called as connective. So this is the site of connective, which is connecting the anther to the filament. Now these 2 lobes of the anther, they connectively show 4 chambers like this. In one lobe there are 2 chambers, so in total there are 4 chambers. These chambers are called as pollen sacs, or these are also called as Microsporangium. So in an angiospermic anther, there are a total of 4 microsporangia present in single anther, or you can also call it as, this microsporangium is also called as pollen sac. Rest of the part is called as anther wall, and this anther wall is not a single layer. It is made up of 4 different parts. Let us discuss one by one. The outermost part of the anther is protective in function, and this part is called as epidermis. Like in all anatomical concept, the outermost protective structure is called as epidermis. Here it is also called as epidermis, which is made up of thin and flattened cells. Stretched and flattened cells are present in the epidermis. It is protective in function. Now next to epidermis, there is one more layer, and that is very important and peculiar layer. You have to remember it. Epidermis is followed by a layer which is called as endothecium. And this endothecium is also called as a fibrous layer. It is called as fibrous layer because this layer shows characteristic deposition of a substance which is called as callose. And this callose is hygroscopic in nature. It means this layer, or this hygroscopic layer of callose, or the endothecium fibrous layer, it will absorb the moisture and swell in size. Due to swelling in size, it will exert the pressure on the anther wall, and the anther wall rupture at maturity to release the spores or microspores or pollen grain which are present inside, that will be released towards the outer side. So endothecium is involved in the process which is called as dehiscence of the anther. This part is very important, and deposition of callus is immensely important for your MCQ kind of questions. So there are two layers. Outermost is called as epidermis. Next one is called as endothecium. The next layer beyond, inside the endothecium, is called as middle layer. And this middle layer is not a single layer. It is made up of 2 to 3 layers of the parenchyma cell. This layer is called as middle layers. The middle layers, they are 2 to 3 parenchymatous layers which are present next to endothecium. They get degenerate at maturity. So in one sense you can understand that there is no particular function of the middle layer. They will just get degenerate at the time of maturity. But remember, at the maturity, endothecium absorbs the moisture to swell in size and rupture the anther wall. That moisture which is to be absorbed by the endothecium is released by middle layer at maturity. So they are also involved in the process of having a particular function in the dehiscence of anther. But when in exams they will ask you what is the exact layer which is involved in dehiscence of anther, then your answer is endothecium, because it consists of the callus which is hygroscopic in nature. The third layer is called as middle layer. They will get degenerate at maturity. The last layer is exactly related with or closely associated with the structure which is called as microsporangium, and that is called as tapetum. This tapetum is nutritive in function. It means it used to secrete certain substances. Now cells present in the tapetum are very peculiar. They show presence of diploid and polyploid cells. And these cells used to secrete variety of substances. It means they used to secrete different enzymes. Then phytohormones are secreted by this layer. Again, they used to secrete the toughest biochemical compound which is called as sporopollenin. So in this way, the anther wall is made up of 4 different layers. One is called as epidermis. It is protective in function. Endothecium is involved in dehiscence of anther. Middle layer, they will degenerate at maturity. Tapetum is the innermost layer which is in contact with microsporangium and is having diploid and polyploid cell. Nutritive in function. It will provide all the nutrition for developing microspore inside the microsporangium, as it is secreting enzymes, hormones and many other substances including sporopollenin. This is the structural aspect of the anther wall. Now this diagram of undehisced angiospermic anther can be asked for 2 marks in your board exams, and other aspects are very important for your NEET examination. Let us summarize the structure of anther wall. It shows presence of 4 different layers. Outermost is called as epidermis. It is protective in function. Endothecium is the second layer. It is related with dehiscence of the anther. It shows presence of characteristic deposition of callus. The third one is called as middle layer. It is 1 to 3 layers of parenchyma and they degenerate at maturity. And last one is called as tapetum. This is nutritive in function, having diploid and polyploid cell, secreting variety of substances like enzyme, hormones and nutritive substances. Along with that, it also shows secretion of sporopollenin. Now this sporopollenin is the toughest biochemical compound which is secreted by the special type of granules, the special type of bodies which are present in the tapetum, and the name of those bodies are Yubish bodies, or you can also call it as Yubish granules. This term is very important for your MCQ kind of questions. The Yubish bodies or Yubish granules, those are present in tapetum. They used to secrete this sporopollenin. This toughest biochemical compound forms the tough layer around the pollen. So this is the pollen gland, and the outermost coating of the pollen shows presence of sporopollenin, and it makes the pollen hard and non-degradable, and therefore pollens are stored for a long time even in fossils as it is. In this way, we have studied the site of microsporogenesis. Now let us discuss what is microsporogenesis. It is very simple. The definition is very easy. The process of formation of microspore is called as microsporogenesis. So microspore and genesis means formation. So you can write in a form like the process of formation of microspores. These are obviously haploid. Now when haploid cells are formed, that process is called as meiosis. So by means of meiosis it is very clear. And haploid cells after meiosis they are formed from the diploid cell. And here the source is microspore mother cell. This microspore mother cell is diploid. So the definition of microsporogenesis is formation of haploid microspores from diploid microspore mother cell by meiosis. This is called as microsporogenesis. Now, where the microspore mother cells are present, they are present inside the microsporangium, the inner pollen sac, which are present in the anther. Now see, meiosis is having two successive steps, meiosis 1 and meiosis 2. So how the process is there, we can explain. This is called as MMC, that is nothing but microspore mother cell, which is diploid. It will undergo the meiosis first. After first meiotic division, it will divide into two cells. Both these cells are haploid. Then each cell will undergo the second phase of meiosis, and that is meiosis 2. Each will produce two cells. And all the cells are haploid because meiosis second is exactly similar to that of mitosis. You have studied this concept in the cell division and cell cycle of the last year's syllabus. So in this way, single microspore mother cell will divide by meiosis to produce 4 haploid microspore. So number of microspore produced from single microspore mother cell is 4. Now these microspores are not separated immediately. Those are form a contact, or you can say form a group, and that group is called as tetrad. Tetrad means group of 4. Now these 4 haploid microspore are present in a specific fashion, a specific manner, which is called as tetrahedral tetrad. Now this tetrahedral tetrad will appear like this. Spore number 1, spore number 2, spore number 3. Where is the spore number 4? That is present behind it. So here you can consider the example of the Brahmadev, which is having 4 heads. You can observe only 3 in a mythological serial. So 1, 2 and 3. Where is the 4th one? It is situated behind. So this particular arrangement is called as tetrahedral tetrad. And these spores, microspores, they are connected with each other in the form of tetrahedral tetrad due to the specific thickening of callos. So this is again very important. Microspore in a tetrad, they are connected with each other by means of callos. So this is the entire process of microsporogenesis, which is very easy, and the type of tetrad which is seen in case of microspore is important. It is present in the form of tetrahedral tetrad. Spores are connected with each other by means of callos.